Thank you, Nicole. To begin with, I'm going to tell you a little bit about the FirePlex technology platform, which we use to do all of these biomarker studies, and then I'll take you through the experimental design and then the results. So the FirePlex technology platform was developed approximately 10 years ago at MIT, and it's based on the optical liquid stamping of hydrogel particles. And when this platform was created, it was specifically designed to be a multiplex platform for doing biomarker discovery and verification studies. And the platform as it stands now is designed to allow you to do multiplex detection of both microRNA and protein biomarkers directly from either tissues or biofluid samples without doing any sample processing or RNA isolation or anything like that up front. And the most important aspect of the platform is that it requires very small inputs of both biofluid and tissue samples. So it's very well designed for studies where you have limited samples and you're splitting it over multiple studies and you really need to get as much information out of it as you can. So the core of the FirePlex technology are these particles that you see here. And the particles themselves are these rod-shaped porous platforms that we use to do all of our Analyte detection. The particles have 3 functional regions that are separated by 2 spacer regions, and I'm going to take you through the architecture of the particles just to explain how do we do our multiplexing and our Analyte detection. So on either end of the particle, indicated in this slide in gray, we have the coding regions, and within these coding regions, we include different levels of yellow fluorescent dyes that allow us to do our barcoding and multiplex detection. In each batch of particles that we generate, we can look at up to 70 unique analytes because we can generate 70 unique code combinations. The center region of the particles, which is indicated here in red, is the Analyte detection region. So for the microRNA platform, we conjugate DNA probes against the microRNA of interest. And for the immunoassay platform, we conjugate capture antibodies against the protein analyte of interest. Now, the particles themselves have several advantages over other bead-based assays based on the design and architecture that we use. The first is that they're made out of PEG, which is naturally porous. So rather than just getting capture on the surface like you do with other spherical beads, you actually get binding all the way throughout the particles. And this provides us with a very large capture surface for analyte detection. And so we routinely see about 5 logs of dynamic range in the assays that we develop. And the PEG itself, because it's biologically inert, you don't see any matrix effects with the other proteins and lipids that are present in biological samples. And so we can work directly with these sample types without having to do any sample processing. In addition, the 2 spacer regions that separate out the functional regions of the particles really do a great job of separating preventing interference and code bleed. So it's a really nice way of doing clean analyte detection. Finally, the assays are designed to be both multiplex and high throughput. So our standard assays are run in 96-well plate format, and we also offer assays that are run in 384-well plate format as well. So in each run, you can look at up to 70 unique analytes per well and up to 90 unique samples per plate. So you generate over 7,000 data points on each plate that you run, which is very important for the samples that we're looking at. The readout for the assays is done on standard benchtop flow cytometers, so it doesn't require any specific or fancy equipment, just a single laser and 3 detection channels. And then all of the data analysis is done with our integrated analysis workbench software. So to show you what that looks like, when the raw data comes off of the flow cytometer, we plug it into our software, of which I'm showing you a screenshot here. And the software does all of the upfront data analysis. So all of the particle decoding, analyte quantitation, statistical analysis, all of the case versus control comparisons, the patient groupings, all of that can be done within the software itself. The software is completely free and it runs on Java, so it can be run on as many computers as is needed. And all of the data analysis that I'm going to take you through was done using our software. So about 2 years ago, I approached our alliance manager, Sandy Chow, who was working with the Michael J. Fox Foundation, and I really wanted to learn more about some of the sample types that the Michael J. Fox Foundation uses for their biomarker studies to try and understand the challenges that are faced, particularly when you're working with things like CSF. And from that and a conversation with Nicole and other colleagues, it spiraled into a much larger project, which is why we're standing in front of you today. And so we set out to actually take on a proper investigative study to utilize the FirePlex technology platform to see if we could find biomarkers of neuroinflammation. And what we really wanted to try and do was to take as holistic and unbiased an approach as we could to looking at these biomarkers of neuroinflammation. So before I tell you about the study design, Nicole is going to come back to tell you a little bit about why it was important to try to take on this broad view.
Great. So we know inflammation and Parkinson's disease are probably linked in some way or another, but we still have some outstanding questions that we need to understand and cover before we can probably make some progress on this issue. So first, is inflammation a cause or a consequence of Parkinson's disease? Is it starting a cascade in the body that leads to pathology and degeneration, or is it an artifact of that process that's already been going on and a readout of that? Or is it both? Is there an interplay between those two aspects? Also, what is the role of central versus peripheral inflammation? There's a lot of studies going on right now with the microbiome in the gut and how that may relate to Parkinson's disease pathology. We see a possibility of synuclein aggregation in the gut, and maybe that's an early readout of future Parkinson's disease. So is there something that's going on peripherally, maybe through the gut, that's causing an upregulation of cytokines, of these T cells, macrophages, that are leading to central nervous system inflammation? Or is it something that is coming from the top down as well, like aging in the brain, genetic mutations, or dysregulation of gene expression in the brain that's driving pathology the other way? Or is it a synergistic effect between these two? So another question is, should we be looking at chronic versus acute inflammation markers? Parkinson's disease is a long duration disease. It's thought that pathology may start decades before motor symptoms show up and diagnosis is made. So should we be looking at things that may be occurring over time and more drawn out, maybe chronic inflammation rather than acute inflammation that's important for other disease spaces? Or should we be looking at both? So what's the role of chemokines versus cytokines in this disease? So if we can tease that out, hopefully we can have a better kind of readout or inflammation profile for Parkinson's. In addition, previous work on neuroinflammation in Parkinson's disease to try to develop a biomarker has indicated that there are some other challenges that we need to kind of work through as well. So there are discrepancies in the published literature. Most studies report differences between healthy control and Parkinson's disease in inflammation readouts. But there are disagreements in the direction of this change. Some studies report an increase with Parkinson's, some report a decrease, some report no change. Some examples of this are interferon gamma and serum TNF as well. There have been multiple studies on these, but there's no consensus on what's changing in Parkinson's disease for these readouts. And this may be due to differences in disease severity of the patients that have been sampled, other comorbidities that are present, different protocols, individual variability, differences in sample processing, analytical methodologies, differences in sampling times, coupled with diurnal fluctuations of inflammatory proteins. The list goes on and on. So it's important to understand that all of these factors may be leading to the readouts that we have. So if we can tease this out, maybe by having very robust, stringent sampling protocols, standardized collection methods and storage methods, maybe that can help us kind of tease this out a little further. And then this is just a figure of a couple analytes that either vary across the time of day or across PD versus healthy control. So you can see that in kind of the tan color, these differ between PD and healthy control with little variability over time. In the middle differs both with time and disease state. And then in blue, these vary across time, but not necessarily PD and healthy control. So some of these changes may be masking any of the effects that we may be able to see. So we started working to try and uncover some of these variables by controlling them, and hopefully this can lead to uncovering the immunophenotype of Parkinson's disease by minimizing these background noise. So we worked with Abcam to provide them samples from our cohorts that have been sampled and collected and stored robustly to hopefully minimize some of this.
Wonderful. So then to take you through what the study design was, we decided to take a multi-omic discovery approach to looking at these neuroinflammatory biomarkers. And so what we really wanted to do was to utilize the fact that we could look at genomic and proteomic biomarkers on the same platform to see if there was a possibility that combining multiple analytes together might get us a little bit closer to finding these biomarkers. And so the approach was to use the FirePlex platform and to look at both microRNA and protein biomarkers in matched healthy and PD patient samples and in matched CSF and serum samples. And we also added a second variable of looking at whether the LRRK2 mutational status also affected any of the biomarkers that we identified. And so the reason for this particular approach was that, to the point that Nicole made earlier, it's potentially important to look at different matrices to see if we can find some of these biomarkers. So for example, CSF is not something that is very comfortable for patient collection, and so doing repeated measures is not an easy thing. So if we could find any biomarkers in the serum that might help point us to a neuroinflammation signature, that would certainly be something that we would like to look at as well. So in terms of the study breakdown, for the microRNA biomarker study, the question that we asked was, are there any microRNA biomarkers that can stratify healthy versus PD patient samples? And can we look at the serum and the CSF to see if we can identify any of these microRNAs? So we utilized a panel of 400 microRNAs that were specifically designed based on what was published to be found specifically in circulating biofluids. And this is particularly important because sometimes panels are designed looking at tissue-specific markers, which is wonderful, but if you can't detect those in a circulating biofluid, then they don't really apply to what you're trying to look at for a blood-based or CSF-based biomarker. For the protein biomarker studies, we wanted to really cast a very wide net, as I said, and we wanted to look at inflammatory cytokines and chemokines, and we really didn't want to restrict ourselves in any way to the analytes that we were looking at. And so the question was, can we identify an inflammatory protein signature in PD patient samples? And when we looked at the panels that we had currently in the FirePlex portfolio, or really what was available commercially in other companies, we saw that they weren't as comprehensive as we hoped. So we decided to actually take an assay development approach first, where we designed a specific 70-plex panel with guidance from the scientists at Michael J. Fox to try to include as many of these analytes in there as we could. And so a lot of the work that I'll take you through was this assay development work that our immunoassay team did in Eugene, Oregon, to build and validate a 70-plex human inflammatory panel. And then once we had validated that this worked in multiplex in the sample types that we were interested in, we ran it with the samples that we had gotten from the Michael J. Fox collaboration. And so this is just a breakdown of how the study was set up. So for the immunoassay-based studies, we had a total of 70 patient samples split evenly between healthy and PD patients, and also split nearly evenly between male and female patients. These were matched CSF and serum samples, and of the PD patients, we included a subset with the LRRK2, that were positive for the LRRK2 mutation. For the microRNA profiling studies, we had a slightly smaller cohort of looking at approximately 38 patient samples, again split nearly evenly between healthy and PD and male and female. The one note that I want to make is that we knew going in that doing microRNA profiling from CSF was going to be a significant challenge. Doing genomics-based assays from CSF has proven to be significantly problematic for multiple assay platforms. And so what we found was that from the CSF samples that we had obtained, they were too dilute based on some pilot data to get robust microRNA signatures from it. And so what we decided to do was to pause that portion of the study and to go back and look and see if we could improve the sensitivity of detection in our assay before we ran it. So there won't be any CSF data for the microRNA studies portion of the talk. The other thing that I want to mention is that, as you can appreciate, we were asking a pretty big question from this patient study. So we were trying to address multiple variables of gender, healthy versus PD, and LRRK2 mutational status from this cohort. And so when I take you through the data, what you'll see is that we didn't find anything that was earth-shatteringly statistically significant that was different between healthy and PD. But that wasn't really the objective here. The objective was to look at a discovery approach and have it help us focus our studies so that we knew what subset of biomarkers we should be looking at, and then hopefully expand that to a larger cohort of patients to do some broad-scale screening studies. And hopefully the statistical power of those studies would allow us to identify clinically relevant biomarkers. Okay, so a quick intro into the assay workflows just to familiarize with you with how we do our assay detection. For the microRNA assays, we started directly with the serum or CSF, and we did a sample digest. We then label all of the released microRNAs with universal adapters, and this allows us to do a universal PCR amplification. The reason for doing a PCR amplification is that when you work with such limited sample inputs, in this case 20 microliters of serum and 40 microliters of CSF, the microRNAs that are present in very low abundance are difficult to detect. And so by doing the PCR amplification, we bring the signal up so that it falls within the linear range of the assay. But because it's a standard universal primer set, you reduce any of the amplification biases based on the sequence variations. So the microRNAs are captured onto the particles, we add on these adapters, and we do our PCR amplification. Within the PCR amplification, the product that we generate is biotinylated. We then add a reporter dye that is conjugated to streptavidin and then scan on a flow cytometer. And the readout that we get of the streptavidin conjugated to this red dye is how we do the Analyte quantitation. So this is just a snapshot of typical data that we see with the FirePlex microRNA assays. So this platform has been validated for, as I said, a broad range of challenging sample types, and we expect to see robust signatures from these sample types. And we utilize the same approach when we were doing the studies that we're talking to you about here. For our standard throughput immunoassays, which are run in our 96-well plate format, the workflow is very similar to a multiplex ELISA, where you have capture antibodies that are conjugated to the FirePlex particles. We add these in solution with our sample. The analytes are captured onto the capture antibodies. We then add a detector antibody that is biotinylated and again add the streptavidin conjugated to the reporter dye and do the assay detection. So all of the analysis by flow cytometry and the data analysis in the software is done exactly the same between the microRNA assays and the immunoassays. The really only difference is that for the microRNA assays, the input was either 20 microliters of serum or 40 microliters of CSF if we had run it. And for the immunoassays, it's 12.5 microliters of biofluid. So from less than 40 microliters of sample input, we were able to do both the microRNA and protein profiling studies. Now, as I mentioned, when we started to look at the portfolio of analytes we wanted to look at, we realized that we didn't actually have an assay, or sorry, a panel that had all of these analytes in it. So we decided to combine Abcam's in-house antibody development technology with our ability to develop custom assays. And so for those of you who may be not familiar with Abcam, we have an in-house technology that allows us to develop recombinant monoclonal antibodies. And we do a lot of custom antibody development. And in particular, a lot of the relationship between Abcam and Michael J. Fox to date has been to develop these antibodies for difficult targets and targets that are of particular importance in Parkinson's disease. But more importantly for this talk, we also use that same antibody platform to do antibody pair development for all of the immunoassays that we have in-house. And those antibody pairs fuel both our simple step ELISA kits and matched antibody pair kits, as well as our multiplex immunoassays. So we decided to use that antibody technology, our custom assay development, and combine those together to make a custom 70-plex neuroinflammation panel. And so this is essentially a snapshot of what we did. So the FirePlex immunoassay scientists and the Michael J. Fox scientists came together to come up with a list of about 80 targets that we wanted to include in our panel. Then our team in Eugene, headed up by our principal scientist, Russ Nooner, set out to really do the panel development and validation. So they screened all of these antibody pairs first in small panels, and then finally in the full 70-plex panel to validate that it could be run in multiplex without any interference or crosstalk or anything like that. Once the pair screening and panel development was complete, we did the validation in the biofluid sample types. I'll show you some of that data in a moment. And then when they were ready to go, we ran the samples that we had obtained from the foundation. So this actually ended up being a multi-site global effort that we took on. So as I mentioned, Sandy is our alliance manager, and she was the person who really helped make sure that this project was moving along and we were hitting all of our milestones. And then our custom immunoassay development team in Eugene really did a tremendous job of developing the 70-plex panel in about 6 months. And for anyone who's been involved in multiplex panel development, that is an outstanding accomplishment. It takes a really long time to do pair screening, but we were really excited that we could do that in short turnaround for these studies. We also had our R&D microRNA team who developed the 400-plex panel, and all of the antibodies that were used were developed in our team by our teams either in Hangzhou or Cambridge, UK. So once all the assays were built, they were sent to the FirePlex service lab in Boston, that is headed up by one of our very talented colleagues, Mike Tackett. And his team then ran all of the samples that were provided by Michael J. Fox on the microRNA and protein panels. And of course, there was a huge amount of input from the team at Michael J. Fox as well. So Nicole and Tarina and Anna really helped inform a lot of the panel design. And then we had studies from Italy and Illinois that supported the biofluid sample types that we used. So just to show you a little bit of the performance data for the 70-plex panel, when we developed this, what we wanted to ensure was that we were seeing sensitive detection of all of the analytes, we weren't seeing crosstalk between the antibody pairs, and that we were retaining the dynamic range that we needed because we knew that some of these analytes were going to be present at fairly low concentrations. And so obviously this is a very busy graph, but what it's just meant to show you is that for all of the analytes in the 70-plex panel, we're retaining about 5 logs of dynamic range. So we expected to be able to sensitively detect all of the analytes, and we're not seeing any issues with antibody cross-reactivity or anything like that. Obviously, these are just standard curves, so this is a little bit of a vacuum, but what's going to be more important is to look at these in biological sample types. And I apologize that this legend is a little bit small, but we validated, I will read it to you. I can barely read it, so I can imagine it's hard for you to read. We validated it in all of the sample types that we routinely screen for our FirePlex platform. So this was plasma that was either heparinized or not, serum, urine, CSF, bronchial lavage fluid, and cell culture supernatants. And so the graph that you're seeing, each of the colors represents the different sample types. And what you can see is that we detect the majority of the analytes in all of these sample types. Now, you wouldn't necessarily expect to detect everything in every sample type because it may not be present, but what we were really looking for was robust detection of the targets we expected to detect, and we certainly saw that. So once we saw that and we knew we had validated it with our in-house samples, we felt confident to then move ahead and run the Michael J. Fox Foundation samples with it. All right, so finally, getting into the results, I will begin by taking you through the immunoassay data first. So this is a snapshot of the data output that we saw for the serum immunoassay profiling studies. And for the 70 samples that we looked at and the 70 analytes in each of these samples, we detected 53 of those analytes above the limit of detection. Of the 17 that we could not detect, 6 of them fell outside the upper range of the standard curve, meaning that they were present in too high a concentration, and the remainder fell below the limit of detection. And so that really points to the fact that when you do such a large plex study, sometimes you then have to adjust the sample dilutions and repeat. So that's something that certainly we're going to look at. So we wanted to start by looking at some of the targets that Nicole mentioned earlier and those that we had already sensed that might be involved in PD. And for the majority of them, we expected that they would be upregulated in PD based on the neuroinflammatory component. So when we look at the results, it's a little bit of a mixed bag. What we saw was that for IL-1 beta and IL-2, those were trending in the right direction. However, they were all detected fairly below the limit of quantitation. And so for these would benefit from being repeated at a lower sample dilution. For IL-6 and IL-10, we could detect them above the limit of quantitation. However, we didn't see statistical significance in the data, even though they were trending in the right direction of being upregulated. Again, that suggests that we just need a slightly larger cohort when we go back to repeat these studies. And then finally, for interferon gamma and TNF alpha, we could detect them above the limit of quantitation, but there was no statistical significance in these studies, so really would benefit from a repeat with a larger patient cohort. So this is just a snapshot of some of the analytes that we identified. So the table on the left is looking at PD versus healthy patient samples. And what you can see is that there is a series of targets that are statistically significant in being differentially regulated in PD. but none of these were ones that were necessarily published in the literature previously. So this is something that we would have to certainly investigate the importance of some of these analytes to see what we can identify. Interestingly, when we look at the LRRK2 positive versus negative patient samples, we do see some targets that are also differentially regulated. So again, this would be something that would be really interesting to further investigate. Oops, I'm sorry. We also did look at whether there was a gender-specific component in these samples. So we looked at male versus female samples, again, PD versus healthy. And we do see a couple of targets, although the hemoglobin and the myoglobin are likely some indication of hemolysis may be happening. But in general, there are some targets in there that may be worthwhile to investigate. Looking at the CSF data, we again looked at the number of targets that could be detected above the limit of detection. And of the 70 samples that we looked at and of the 70 analytes in that panel, we could detect 42 of the analytes above the limit of detection. Of those, of the 28 we couldn't detect, 3 fell outside of the upper range of quantitation on the standard curve, and the rest fell below. So the case of CSF was, as we had seen with the microRNA, the samples are just fairly dilute. And so we would need to go back and repeat this, particularly for the targets that we couldn't detect at a lower sample dilution to hopefully bump the signal for all of those back into the linear range of the assay. And again, we took the approach of looking at what was already known in the literature to see if that could help inform some of the data that we were looking at. And what we found was similarly to what we had seen with the serum data, the IL-1 beta, interferon gamma, and TNF alpha are not present in the linear range of the assay. We couldn't quantify them. And so this would really bear repeating at that lower sample dilution that I mentioned. And then for the others, so IL-6 was present and detectable. However, we didn't see any statistical significance when we looked at the data, although I will show you some data on the LRRK2 patients for IL-6. That's a little bit interesting. And then the PGE2 and osteopontin weren't included in this initial panel design, so we can certainly go back and include those in future panel designs as well. So looking at the PD versus healthy patients, we didn't identify any targets in CSF that were statistically significant in being differentially regulated. But there are some really interesting targets that jump out in the LRRK2 positive patient samples, particularly the IL-6, as well as some of these other interleukins. And so given that there was only 10 patient samples that we looked at between the LRRK2 positive and negative patients, this would be something that would be really interesting to further investigate with these larger patient cohorts. So I'm excited to see what may come of that. And it is encouraging to see that it is upregulated in the LRRK2 positive patient samples for IL-6, which is what we expected. And then looking at the CSF data, male versus female samples, we do identify these targets of IL-8 and liver fat B. So I'm not sure what the clinical significance of this would be and whether potentially in CSF there is a potential for a gender-specific signature, but this would be something that we would certainly need to investigate further. And again, this was really an objective of trying to look at all of these analytes to see if we could refine that panel down to a smaller set of targets so that we could do these larger scale patient studies and get some usable data from it. And then very quickly looking at the microRNA data, as I mentioned, we solely focused on the serum for these studies. So for the 36 samples that we looked at, 110 out of the 405 microRNAs that were present in the samples could be detected at a mean fluorescence intensity above 1. And so what you're seeing in this heat map is sort of the spread of microRNA profiles. And so for the majority of them, we're seeing low fold changes. This is a log 10 scale, but then there are some targets that we do see pretty significant fold change differences in. Now, interesting for the microRNA data, while we didn't see any microRNAs that jumped out as being differentially regulated between the LRRK2 positive and negative samples, we did see a couple of microRNAs that were statistically significant between PD versus healthy patients. And what's interesting to note is that miR16 is a neurospecific microRNA that's really robustly detected in neuronal sample types. And so it's interesting to see its enrichment in serum in the PD patient samples, potentially suggesting that there may be some microRNAs that are entering the blood in response to this PD. Again, absolutely bears further investigating. And then looking at the male versus female samples, again, we see some microRNA signatures that are differentially regulated. This was a slightly smaller study, and so I do think that if we expanded the patient cohort size, there would be some additional microRNAs in there that we may identify as being differentially regulated. And what's particularly interesting to me from these studies is that I understand from Nicole that there are collaborators within the Fox Foundation network who are running RNA-seq studies. And so it'd be interesting to use the data from the RNA-seq to inform a panel design that could be more focused on these PD-specific microRNAs and then to repeat these studies again.